Prokaryotic Form and Function, Chapter 3, Topic 3.1. In overview, this topic goes into more detail about the nature of microbes as compared to what we went over in Chapter 1. We're going to talk about the distinguishing characteristics, prokaryotic cell structure, and prokaryotic shapes and arrangements. At the end of this recorded topic lecture, you should be able to meet the following objectives. Name the structures that all bacteria possess. Name 3 structures some, but not all, bacteria possess. Describe 3 major shapes of prokaryotes, and describe 2 major cell arrangements of prokaryotes. Let's talk about distinguishing characteristics of prokaryotes, in other words, characteristics that distinguish them from eukaryotes. These are the way the DNA is packaged, the makeup of the cell wall, and internal structures. So bacteria do not have a nucleus, they do not have a lipid bilayer surrounding the DNA within the cell, and they do not have histones. Histones are proteins that are unique to eukaryotes that the DNA is wrapped around as a part of the packaging process, and we'll talk about that more in later chapters. Moving on to the cell wall, bacteria have peptidoglycan, and archaea have molecules that are unique to that kingdom. They could be pseudopeptidoglycan, they could be straight proteins. I'm not going to make you remember them, okay? But just remember that archaea do not have peptidoglycan, but they also don't have cellulose and chitin and other molecules that are unique to eukaryotic kingdoms. Internal structures. Prokaryotes do not have any membrane-bound organelles. So here's a diagram of prokaryotic cell structure. Yes, I want you to know this for an exam. And yes, this looks different than the picture in your book. I want you to get familiar with picking out these structures in different diagrams. Okay, that's part of critical thinking. When you're first memorizing something, then you memorize it based on the information that you have, but remember, I want you to be able to apply it to novel situations such as diagrams you haven't seen before. So let's take a look at this diagram in more detail. The items that are in red are items or structures that all prokaryotes have. They all have cytoplasm, they all have ribosomes, they all have a plasma membrane, and they all have a nucleoid containing the DNA. Now, the terms in black are things that some bacteria have and some bacteria do not. So some bacteria have inclusions, some of them have pili, some of them have a capsule, yada yada yada. So I'm going to leave you to memorize these terms and the functions of each of these structures on your own in the textbook. Now let's go into the various shapes that prokaryotes can come in. If they're round, we call them cocci. If they're rod-shaped, we call them bacilli. Now, sometimes the bacilli can get so long, they form what I call variations on the rod shape. So, if they form kind of a comma shape, they're vibrio, like in this electron micrograph. If they're long enough, they can form kind of a spiral or a corkscrew, and they're rigid, they're called spirillum. Now, spirilia generally have flagella coming out of one or either end. Not always, but they can. Then if they're long and flexible, they're spirochetes. Now, spirochetes do not have external flagella. Their flagella are kind of embedded, like an embedded ingrown hair, and you won't ever see external flagella on a spirochete. Now let's talk about the arrangements that bacteria can take. When they divide, they separate, and they're always separate circles, we call them cocci. If they tend to form pairs of 2, we call them diplococci. Now, if they divide in just one plane of division, when they divide, they always go this way, always go this way, and they stay together, they form strings or streptococci. So streptococci are strings of spheres or cocci. Now, some bacteria don't like dividing in just one plane of division. They get bored. So they'll divide in this plane, and then the next time they'll divide in this plane. So if they stay together when they do that and they form these cute little squares of 4 cocci, we call them tetrads. In lab, Micrococcus luteus tends to form tetrads, so you'll get a chance to see them. Now, if they stay together to form associations of 8 cells, then they form sarcina, or these cubes. Now, staphylococci divide in several planes, and they form grape-like clusters of cells. Staphylo is Greek for grape-like, or grape-like clusters. Therefore, staphylococci. Now let's talk about the arrangements that rods or bacilli can take. Now, they can be single as well, so we just call them bacilli or single bacilli. If they tend to stay together in groups of 2, we call them diplobacilli. If they tend to stay together, then we call them streptobacilli. Now, you'll notice there are no staphylobacilli. That's because they can't divide in multiple planes. Besides, we have a tendency not to see them in grape-like clusters because grapes don't come in rod-shaped. So, no staphylobacilli. No staphylobacilli. Now, some bacilli are short enough and lumpy enough that right after they divide, they look like cocci. But as they grow, they look like short, fat little rods. So we tend to call them coccobacilli, or coccobacillus. E. coli is a coccobacillus, so you're going to be able to see that in lab. So don't freak out when you're E. coli. Sometimes they look like cocci, sometimes they look like bacilli. That's the nature of E. coli. Well, that's it for this topic. You should now be able to name the structures that all bacteria possess, name 3 structures that some but not all bacteria possess, describe 3 major shapes of prokaryotes, and describe 2 major cell arrangements of prokaryotes.